Bismillahirrahmanirrahim. Assalamu alaikum and a very warm welcome to all of you. It's a real pleasure to see all the students again and also to sit in my own office and be back to our usual business. So as you know, you are in Genetics and Genomics BIO621. And I shared with you a detailed course outline. I shared uh, everything, and I would encourage you to go through the course outline. This course is basically um, about principles of, functional genomics. It's about how genetics tools, methodologies, and genetic analysis can be used to understand gene function. So whatever we'll do, we'll cover from research point of view. You may find many things redundant. You may find many things in today's lecture like um, repetition. But today's lecture is first few lectures, 2-3 lectures are fast-paced to remind you or to refresh your fundamentals, and then we are going to enter into all the functional genomics, the forward genetics, reverse genetics, molecular genetics, which is epigenetics, genetics of development, genetics of flowering, genetics of gene silencing, transposons, RNA as a molecule, gene evolution, etc. You will be given research papers, and it's mandatory. You should read those papers. Whichever papers I share with you, you have to read those papers. As I said, this is a master's level course. In this course, you should not expect that, I should teach you each and everything by holding your hands. I'll cover as much as possible in the lectures, but then I'll give you papers so that you read those papers and try to understand different methodologies, different tools, experimental design, because at the end of the day, you will be designing experiments. So in a year's time from now, you will be designing experiments in the lab, and then also, you will be interpreting your results. So you should make it a point that whichever paper I share with you, you read those papers. Okay. So let me share my slide. So, as a biologist, we aim to understand cell function, and what we dream of is to understand function of each and every individual component within the cell. There are different compartments, organelles within the eukaryotic cell, and all those are controlled. The function of a living cell is actually controlled by the protein molecules which are present within the cell. Now, those just hold on, I'm receiving an email from another student that his link is not working. So in order to understand the function of the cell, we need to understand, function of the different molecules that exist within the cell. And one of the fundamental molecule which acts as a template and regulates more or less every cellular process, or predominantly majority of cellular processes, is the DNA molecule. You all know DNA is composed of, the 4 nucleotides. This DNA molecule is wrapped around specialized proteins which are called histones, and these two components, the DNA and the histone proteins, they are packaged in such a way that the higher order structure of these two molecules gives you a chromosome. And these chromosomes are the fundamental unit of heredity. These chromosomes, they are the fundamental unit of inheritance. So in biology, we have many questions. For example, as biologists, you may be interested in understanding the how you breathe air, how your muscles move, or how you fight back infections, how your body immune system works. So there are unlimited questions. There is unending list of questions. And if you want to understand any of this function, biological function, you have to understand, as I told you, you have to understand the molecules. You have to go at the molecular level to understand which function is encoded by which molecule within the cell. There are two fundamental approaches to understand a biological function. One is gene-based, and the other is protein-based. So gene-based approach is, of course, it is called genetics, and the protein-based approach is called biochemistry. So in this course, we are going to focus on the gene-based approach, which is the genetics. And we are trying to learn as much as possible in next 28 lectures, how can we dissect function, biological function, by employing different genetic strategies. How can we design experiment based on genetic tools, which answers the fundamental question of a specific biological function we are interested in. For example, the question you are interested in is how butterfly flaps its wings. Now, as I said, there are two approaches. One is biochemical approach. The other is gene-based or genetic approach. And biochemical approach is simply you grind the whole butterfly, and all the cells of butterfly, they are dead. All the components of cells, they are in a solution, and you isolate individual components. Basically, as biochemist, you are purifying proteins. There are biochemists which work on carbohydrates as well. There are biochemists which work on lipids as well. But in terms of understanding the biological function, we isolate proteins, we purify individual fractions, and then we try to find component which has a function, muscle-like activity or whatever, which can help fly in its flight while using or flapping the wings. Another independent lab can be interested in how yeast, it utilize sugar. Again, what a biochemist is going to do, or biochemical approach is going to do. It's going to grind yeast, make all the yeast extract which contains proteins, and then find proteins which can digest sugar. So in contrast, the geneticist is actually focusing on keeping the organism alive. It's not going to kill the organism. Rather, it tries to create a mutant. In genetics, we create mutants. Okay, and while creating mutants, what geneticist aims is that, since I'm interested in this question, how butterfly wing development takes place, or fruit fly wing development takes place, or how it can flap its wing. So as geneticist, you will create a fly in which wings are defective, wings are missing, or wings are deformed, and rest of the organism is viable. Similarly, if you are working with a yeast, you will try to make a yeast which is viable, ideally which is viable, but it's defective in utilizing sugar. It cannot utilize sugar, and otherwise it's normal. Okay, so it means you have created a mutant which is carrying some mutation in gene which is responsible for utilization of sugar or sugar metabolism. So this mutant is normal, but this mutant has defect in wing. This yeast otherwise is normal, but this mutant yeast is not able to utilize sugar. So that's what we do in genetics, or the functional genomics as well. However, this distinction between genetics and biochemistry is totally dissolved. It's no more existing now. And the discipline is called molecular biology. As molecular biologists, for example, in my lab, we are basically a genetics and epigenetics lab. We use a lot of gene-based approaches, but we are doing a lot of biochemistry as well, because without doing biochemistry, you cannot understand the cellular function. Even if you clone gene, you make a mutant, you have a mutant, you have to clone the gene, express the protein, and characterize that and find where it goes. So we do in molecular biology, you do genetics, you do biochemistry, a lot of cell biology, and so on. Okay. So I'll take you 100 years back, and in the beginning of last century, when they rediscovered Mendel, they came up with a fundamental question. And the fundamental question was, because by the time Mendel discovered his laws, he had no idea about genes, chromosomes, relationship between genes and chromosomes. So in the beginning of last century, T.H. Morgan's lab it introduced fruit flies in the labs. And all the work by T.H. Morgan, it went to formulate a theory which is called the chromosomal theory of inheritance. According to chromosomal theory of inheritance, the genes, or formally what Mendel said, factors, they actually reside physically, they are associated with chromosomes. And these chromosomes, they are the ones which actually inherit from parent cells to the daughter cells. So whatever the genetic makeup of a living cell is, or the genotype of a cell is, and the corresponding phenotype. So genotype and the corresponding phenotype, they are inherited through cell division. And this inheritance of phenotype is actually due to this association of genes with the chromosomes. I would like to quickly remind you that Mendel was rediscovered because of discovery of meiosis, because of discovery of chromosomes. In 1904 or 1905, I believe, they were just visualizing cells under microscope, and they realized that, you know, there are cells from same organism which contain 4 chromosomes, and then there are cells from the same organism which contain 2 chromosomes. And the cells which contain 2 chromosomes, they were very few or selected as compared to majority of the cells. And this is actually meiosis. So you have diploid cell and you have haploid cell. So what they discovered that, you know, these are specialized cells which we refer today as germ cells, and these are somatic cells, and, you know, actually division, cell division, a specialized cell division which is called meiosis, that leads to formation of gametes, and these gametes from male one end and female one end, they restore the diploid status of an organism. And that's how, you know, an organism grows, because now from here it divides mitotically and always produce 2n somatic cells. So by looking under microscope and finding that there is difference between the number of chromosomes in cells of same organism, and, you know, that is due to specialized meiotic cell division, and then the others are mitosis. That was the rediscovery of the Mendel. They discovered that, okay, what Mendel was referred to as the factors, okay, and he said only half of the factor goes to the next progeny. Actually, this is, Mendel was assuming meiosis, and, you know, those factors, they must be present on these entities which they call chromosomes. Chromosomes because chrome mean color and some means something. So chromosomes under microscope, they're just, you know, stained with a dye, so something that can be colored. So with that, I just remind you what Mendel discovered. So a quick refresher. So what Mendel showed us, he studied 7 different traits, and, you know, let's take a trait called seed shape here, and he took round, crossed it with wrinkled, he got all rounds, He self-crossed F1, and what he found was rounds versus wrinkled, the ratio was 3:1. It was like, 5,400 something, and then let's say, uh, 1,000 X, Y, Z. So the ratio turned out to be 3:1. Okay. Now, what he did, he assumed, and he said, okay, since forget about these two capital R's at the moment, just focus on these phenotypes I have written. Once he realized that round dominated the wrinkle completely disappeared from the F1, he said, okay, round is dominant and wrinkle came back only in F2, said that wrinkled is recessive and round is dominant. And then he started assuming that the dominant trait I'm going to designate capital letters, capital R, and the recessive one I will designate with small r. He assumed that, each of the parent contains these factors. They exist in pairs. Since this is recessive, small r, but each factor, each parent contributes only half of the factor, and that's why F1 looks like this. You have F1 self-crossed, and you have 3:1 rounds. They were like this. So 3:1 was basically the phenotypic ratio. But he also discovered that if he randomly takes F2, if he randomly takes F2 plants, seeds, and then grows them further, what he finds that, let's say out of 100 F2 independent randomly selected seeds, he found that out of 100, there is a ratio which he called 1:2:1. One was pure breeding round, two were impure rounds, and one was pure wrinkled. Pure means there is no contamination. Pure means it will always produce wrinkled. Pure means this will always produce round. This is impure because when you will sow seeds from this plant, this will again give you 3:1. Okay? This is undergrad genetics. Now, he did such experiments with many different traits. For example, he took yellow seeded peas with green seeds plants. He found all F1 is yellow. And when he self-crossed yellow, he finds in F2 yellow green was again 3:1 ratio and 1:2:1 genotypic ratio. So this is the genotypic ratio where you are defining at the level of genotype, and this is where you just look at phenotypes, yellow versus green. These are phenotypic ratio. And he found that, okay, I can fill in again factors. I will, the dominant trait, dominant is yellow because all F1 is yellow. I'll designate capital Y, and recessive trait, which is green, I'll designate small y. Now I assume each parent, so factors exist in pairs. Each parent contributes half. So in F1, you will be like this. You can make gametes like this. Okay. It's random fusion. So this will be F1. If you self-cross F1, you get 3 rounds, which will be looking like this, and 1 green, which looks like this. This is 3:1 yellow green, and this one is 1:2:1 genotypic ratio. He did these things with 7 different traits, and he also started predicting that, because whatever he found, it was true. So he started predicting, and he said, if I use F1 heterozygous and cross it with recessive parent, I should get 1:1 ratio. Okay. F1 heterozygous was what? This thing. And homozygous recessive was this thing. What will be the ratio? You make gametes, you get ratio 1:1. This is called test cross. Whenever you cross F1 heterozygous with homozygous recessive parent, that is also called test cross. Is it clear up till now?
Yes, sir.
Okay. Now, this is all up until this thing. We are talking about monohybrid cross. Okay. Why monohybrid? Mono means one. Hybrid means, when you have 2 different genotypes, 2 different parents are crossed, and progeny contains genome or genes from 2 different parents. Okay. Mendel also did this with the next step he did. He said, okay, let's do something and combine them now. I had yellow, and I will forget about this. I take yellow. Always write phenotypes first. Don't go to genotypes. Yellow wrinkled crossed with green rounds. What I will get? What he got yellow rounds. He self-crossed them to see what is he getting in F2. The whole idea was he wanted to see if 2 different factors, when I have 2 different factors within the same parent, if they are interfering with each other's inheritance. And in F2, he found phenotypic ratio, which is famously known as 9:3:3:1 phenotypic ratio. So 9 were yellow round, 3 were yellow wrinkled, 3 were green rounds, and 1 was green wrinkled. Okay. Now let's add the genotypes. Yellow was, we know already, capital Y, wrinkle. Green was small y, and round was like this. You make gametes, and in F1 he received like this. And when he looked at F2, he received, for example, yellow rounds. There was something like, let's say, 308 or so. He had yellow, wrinkled, you know, 108 or so. Sorry, green rounds. Again, he received something like 400 something, and he had green, sorry, not 400, 100 something. Green wrinkled were like 25 or whatever numbers. These are just random numbers I'm throwing. Now, when he calculated, he found the ratio between these phenotypes is 9, 3, 3, and 1. And what he did, he tried to see if the yellow or green, it is interfering with round or wrinkled. What he did, he simply forgot about green and yellow. He just ignored green and yellow here. He said, okay, let's add up all the rounds. So where are rounds? You have, this round plus this round, 308 plus 100 x. X is just a number. Recessive for round is wrinkled. What was wrinkled? 108 plus whatever, A plus B, he got Y. Okay. When he divided, he found that the ratio is 3:1. The ratio of round to wrinkled is not disturbed even in a dihybrid cross. He did same for yellow versus wrinkled. He forgot about round. He added all the yellows. Let's say M plus N is Z, and all the wrinkled. So here are wrinkled this one and this one. Let's say this is C. This is C plus D. And again, he found that that ratio of yellow wrinkle was also 3:1. So the discovery Mendel had was that underlying 9331, there are two hidden 3:1 ratios. One was round versus wrinkle, the other was yellow versus green. So this is what you know as independent assortment or whatever. But as I showed you, Mendel was rediscovered due to discovery of meiosis. So what they next did was that after discovery of meiosis and predictions that what Mendel discovered, it was not useless discovery. It makes sense. So Morgan's lab at Columbia, they were working with fruit flies. Morgan was the first. T.H. Morgan was the first person who introduced actually fruit flies in the lab. Peas were long generation organism, maybe 6 weeks to 8 weeks. Flies were 10 days. In 10 days, fruit flies life cycle is completed from egg laying all the way to adult hatching. And you can go to next generation. So starting a cross, then next generation F2, F3, you can just add up 10 days. So what Mendel's lab discovered that they have fruit fly mutants, and they have they discovered fruit fly whose body color was black. When they crossed black body color with wild type fruit flies, all the F1 was wild type. Okay, it's a invertebrate, and it's not like plants where you can do self-crossing, but you can do crosses between siblings. And when they crossed, so by wild type here, let me simplify. The body color of wild type fly is brown. Okay. Now all the F1s were brown. When he crossed F1 brown with F1 brown, they found, numbers, let's say 500 browns versus 160 XYZ, whatever numbers, as black. So what do you find here? What is black versus brown? Minahil?
Yes, sir, it's almost 3:1.
Yeah, but when you look at phenotypes,
Yes, sir.
black body color versus brown, what do you
So
conclude?
brown one is dominant and black is recessive.
Good. So brown is dominant, wild type is dominant, and black is recessive. So I'll write here brown, let's say, and the black with small b, small b. Remember, you can also use for wild type like plus plus, or it's your call. You can also write b plus like this, and black can be written like b minus, but my advice to you is go with capital and small letters. Okay, but if you find somewhere written plus plus, it means two copies, homozygous copies of black brown gene. Okay, so what they did now, following Mendel's method, the F1 is this. When F1 brown is crossed with F1, what you will get? You will get which is almost 3:1. Okay. They found many traits, many phenotypes in fruit flies. Let's say another one, which is in the slides, is they found flies which are called the phenotype. They named the phenotype because wing was deformed. They named the phenotype vestigial. They crossed vestigial flies, and in vestigial, the phenotypes are totally gone. The wings are totally gone. They crossed with the normal wings fly, normal wild type fly. They found in F1 normal wings. They crossed F1 normal with F1 normal, and they again found 480 normal, and vestigial something like 105, let's say. again like near 3:1. This shows you vestigial is dominant.
Sir,
Taha?
vestigial is recessive.
Yes, vestigial is recessive. Okay, so normal wings, let's say capital VG, and vestigial is small vg because it's recessive. So you add up these things. Small vg, F1 will be like this, and in F2 you will get 3:1 or 1:2:1, the Mendelian ratios. Okay, but they were getting strange numbers. So as monohybrid they were fine. These are monohybrid crosses, isn't it?
He said they are monohybrid.
These are monohybrid crosses. But what they discovered that there's, whenever they used why it's coming, my slides, why my slides, I had it so many white slides. Okay, let's use this slide. Whenever they used black vestigial cross to normal flies, which means brown and normal wings, whenever they try to do a dihybrid cross, they did get normal flies, which means brown body color and normal wings. Okay, but when they crossed these flies with F1 normals, the F2 ratios were not precisely 9:3:1. Okay, F2 ratios were not precisely 9:3:1. There was a problem there. In Mendelian case, what you expect? Let's write it down. Black vestigial will be what? Small b, small b. Normal will be, and this normal will be when you cross it with a sibling of the same genotype, you expect this ratio, but this was not the case. What they did, they said, and for many years, postdocs working in Morgan's lab, they could not figure out what is happening there. None of the postdocs, wanted this project. And a sophomore student, Sturtevant, joined in. And what he did, he said, let's take black vestigial, black normal wings, and cross it with brown vestigial. What he get? What he expects? What he will get? Hassan Sajjad?
Yes, sir. He'll expect 9331.
No.
Sorry. He will expect normal ratio, jo, sir.
No, when you
F1.
cross black body color normal wings with brown body color vestigial flies, what
Yes.
will be F1?
It will be. No idea, sir.
Ji Taha?
Yes, sir.
If you cross black body color and normal wings fly with brown body color and vestigial fly, these are two different parents.
Yes, sir.
What will be F1 phenotype?
Sir, brown normal wings.
Brown normal wing. Excellent. Because brown is dominant over black and normal wings are dominant over vestigial. So it will be normal fly. Normal. What is normal? So black is small b. Small b normal wings are, we said, like this. And brown is capital B, and vestigial wingless flies were like this. So in here you will get like this. He said, I'm going to cross, Stratton said, I'm going to set up a test cross. Because people were not able to get these 9:3:1 ratios so precisely. Said, okay, I'm going to get, a test cross, and test cross is what? Ji Ahmed Naqvi, test cross kya hoga?
सर वो आप रिसेसिव से क्रॉस करते हैं ना?
हाँ जी, very good. So you will cross with homozygous recessive. ये homozygous recessive होगी. The beauty of having test cross is you forget about the gametes from this side because you know it's homozygous recessive. And Stratton said, okay, what I want to see is ratio between parentals and non-parentals. What is parent? This is parent. And this is parent. Whenever we will see, we will say original parental, not the F1 parent. F1 we know is heterozygous. We are talking about original true breeding parent we started with. And the parental gametes, what were the parental gametes? Parental gametes are B, VG, and second parent was capital B, small vg. But in Mendelian case, you remember there were always non-parentals as well. When you cross yellow round with green wrinkled, what did you get in F2? You get yellow rounds, you get yellow wrinkled, you get green rounds, and you get green wrinkled. You remember? So in Mendelian case, so if yellow round was parental 1, green wrinkled was parental 2, which means parentals in case of Mendel are capital Y, capital I, or small y, small I. Okay. It can be capital Y, small I as well if you had yellow wrinkled as parent. Okay. But these are non-parentals, which means capital Y, small r, and small y, capital R. So study one, because they were getting in their this cross, in these crosses, they were getting these non-parentals as well, but something was wrong there. So he said, okay, I'll see what happens here. And the non-parentals, so B, what will be non-parental one? Capital B with capital VG, and the other non-parental will be B with small vg. So I write down these two are non-parentals and these two are parentals. What do you expect according to Mendel? According to Mendel, let's also write down Mendel as well here. Let's say we started parentals in case of Mendel was yellow round, green wrinkled. So non-parentals will be yellow wrinkled. And according to Mendel, what do you expect? What is the ratio of each one of them? The gametes?
9331.
Huh?
9331.
9331 is the phenotypic ratio. Here, we are talking about gametic ratio. We are ignoring here at the moment this one. In case of Mendel, it's always 25% each. 1:1:1:1 gametes. They were not getting 1:1:1 ratios. Okay. So since the discovery of meiosis, an idea was there. They had learned that the genes, they associate with these chromosomes, and they were getting strange numbers. They postulated a question, and the question was, can it be that Mendel's yellow and round or wrinkled, they could be on different chromosomes? And what we are working on, black and vestigial, they could be on same chromosome. This is what they postulated. And they said, if genes are on independent chromosomes, we get 25% each, or 1:1:1 ratio, which eventually results in 9:3:1 ratio. What if Mendel's both the traits were on the same chromosome? What? Instead of being on independent chromosomes, if both the traits were on the same chromosome, according to Mendel, factors, they exist in pairs. Each parent can contribute only half. So this should go to one gamete. This should go to other gamete in F1. And the other parent should contribute small y, small r. So F1 will be what? In case of Mendel, if we draw chromosomes, F1 will look like this. When you will cross F1, according to Mendel, now these chromosomes, they inherit independent of each other. The factors inherit independent of each other. So in this case, when the gametes will form of F1, where's my hand? So one gamete will contain this one, this chromosome, the other will contain this chromosome, and same for this and this. So you should get, if they're on the same chromosome, you should get 50%. Clear? If they're on the same chromosome. So Morgan's lab assumed that, let's test this hypothesis, because our numbers, they don't fit in classical 9331 or the gametic ratio 25% each. What if genes are on the same chromosomes? And because everything they were doing, they tried to prove Mendel, and in monohybrid crosses and in many dihybrid crosses, I should tell you, in many dihybrid crosses, they could reproduce Mendel as well. But these numbers did not fit well. And what they discovered that, there are, for example, here, let me go to the slide now. I have this slide here. All these slides will be on LMS. So you should go and, here. No. Yeah, this is what we were just talking about. And black vestigial, these are the numbers you expect. Parentals, plus means plus for, this is brown, you can assume it B, and this one is capital VG, okay? Like this, homozygous, okay? So here plus means capital B, and here plus means like. So this is what they were trying to see. And what they discovered, that the numbers they found, they were like 965, 944 for parentals, and non-parentals were 206, 185. Much smaller number. It was neither 50-50 and it was nor 25% each. This is the test cross they set up. Strativan set up the test cross. What Strativan did next was, he added up these numbers. He said, okay, I'm going to add all the parentals. But one thing is very important to note here. The ratio between parentals is 1:1, and ratio between non-parentals is also 1:1. In two separate categories, parental show 1:1 and non-parental show 1:1. Okay, so Strativan did very intelligent thing, and he said, okay, I'm going to add the non-parentals, which are these ones, 206 plus 185. 206 plus 185. I'm going to add non-parentals and divide by total and calculate the percentage. So 17% were non-parentals. Since he used, females like this and males like this, this is the sign of male, this is sign of female. He said, okay, I do a reciprocal cross, I change the parents. So one parent is homozygous recessive for vestigial, wild type for body color. The father is homozygous recessive for black and normal wings. And he said, okay, I get F1. I'll go to set up the test cross. And after setting the test cross, I will again look at parentals and non-parental combinations. Maybe, you know, coming from different parents, it disturbs. What you find with little variation, again, same numbers. And again, if you add up these numbers, non-parentals divided by a total into 100, roughly same percentage. And he said, these two genes, they reside on the same chromosome, because they could then easily figure out. And when genes reside on the same chromosome, Mendel's law of independent assortment will not hold true, because these genes, they will depend on recombination. Genes which reside on the same chromosome, they are referred to as linked genes. Genes which reside on the same chromosome, they are also referred to as linked genes. Okay. Now, he named these non-parentals. He said these are called new combinations. And from here, the term evolved, which is called recombination or recombinants. What happens? You have, you know, this is your F1. Okay. You have these chromosomal makeup. This is F1. You remember this one? Heterozygous with normal phenotype. In chromosomal form, you can draw them like this. These are heterozygous. Okay. When they will enter into meiosis, DNA replication will take place. Both the homolog chromosomes will have sister chromatids. So this is one homolog sister chromatid. This is the other linked at the centromeres. Okay. मैंने गलत जगह पे centromere draw कर दिया. Centromere कहीं side पे होना चाहिए था. ठीक है? Centromere should not be here. Centromere should be far away. But I just drew this centromere just to show that these are sister chromatids. Okay. And this is one homolog chromosome. This is the other homolog chromosome. Now, based on the distance between the two loci, position of a gene on chromosome is called locus. Locus. Plural is loci. Position, so distance between the two loci, black, capital B, and vestigial, that determines this ratio of recombination. So bigger the distance between the two loci, higher the frequency of recombination. So these numbers, non-parentals, they depend on how farther apart these two genes are from each other on the same chromosome. And during meiosis, they look like, you know, like this. Okay, two homologs come together. इनको मैं chromosomal form दे रहा हूं. This is crossing over taking place during meiosis, and this structure is called chiasmata. So based on the distance, maybe black is there, vestigial is there. Okay, and they are getting into, so you get 965 parentals, 944, for example, another parental, and these non-parentals combined together are 17%. Clear? So this led to a conclusion that when genes reside on the same chromosome, based on the distance between, physical distance between the two chromosomes, there will be certain percentage of recombinants. It will never be 25% each, or 1:1:1:1 ratio, which Mendel was getting when genes reside on the same chromosome. They do inherit independently. It's true. Independent assortment is true. Inheritance of one gene does not disturb other, but when they are on the same chromosome, they may go together. So I think the class time is over. कितने बजे तक ये class होनी है?
So 10:45 तक.
Yeah. So these slides are on the LMS, and all what I explained you, or all what I talked in the beginning on the blank slides, all this. So all what I talked here is actually here in these slides. These slides are just helping material. Don't just restrict yourself to these slides. Go back and solve, I would say, monohybrid, dihybrid, some recombinants, problems. In next lecture, when I talk, I'll talk about recombination more in detail, and we'll give you assignments as well, so that you practice. Okay. So the more you practice, the better it is. Any questions? Are there any questions? So I think
Yes,
today's,
sir.
yeah.
सर डिस्टेंस बिटवीन द टू लोसाई जितना ज्यादा होगा फ्रीक्वेंसी ऑफ रिकॉम्बिनेशन
उतनी ज्यादा होगी.
उतनी ज्यादा होगी. ऑलराइट.
Yeah, if the two genes are very close to each other, recombination becomes 0, and that's where we say linked. Genes are linked. Genes which always inherit together during meiosis, which means they are not undergoing any recombination, they are called linked genes. And based on the recombination percentage, मैंने अभी screen share करना बंद कर दी है. Let me just किसी की class तो नहीं है next? So based on the recombination percentage, let's say 17%, you should know that was the discovery of unit they named. They said, okay. So they said that frequency of recombination, maximum frequency कितनी हो सकती है? मैथ तो पढ़ा हुआ है.
100
परसेंटेज होती है 100%. रिकॉम्बिनेशन फ्रीक्वेंसी मैक्सिमम 1 होती है, जहां तक मेरा पुअर मैथ. ठीक है. यू विल गेट 0.17 or 17% recombination. They said that if there are 100 meiotic events, शकूफा आपने क्लास लिया, आपको ये पता होना चाहिए. If there are 100 meiotic events, and if we find 17 recombinants out of 100 meiotic events, if we find 17 recombinants, the distance between black and vestigial is 17 centimorgan. Centimeter से नहीं होता, it's a distance. So they honored their mentor by adding Morgan's name there. So let's say you get recombination, you are working on a gene called A and gene B. You did analysis, all these things, and you know parental, non-parental, dihybrid cross आपने set किया था. Non-parentals, इसमें parentals कौन से होंगे? Sorry, F1 आ गया like this, and then we set a test cross and F2. इसमें parentals कौन से होंगे, non-parentals कौन से होंगे? उस्मान, आवाज नहीं आ रही, आपका माइक म्यूट है.
स्मॉल ए कैपिटल बी बी ये पेरेंटल होंगे और F1 में जो आएंगी जनरेशन वो नॉन पेरेंटल में आएंगी हमारे पास.
दोबारा बोलें प्लीज, पेरेंटल क्या होगा?
पेरेंटल हमारे पास स्मॉल ए कैपिटल बी बी जो है ये पेरेंटल में आएंगे और जो F1 जनरेशन में आएंगे वो नॉन पेरेंटल में.
नहीं, आप मुझे, अगर हमने टेस्ट क्रॉस सेटअप की है, ठीक है, और हमने
Yes,
ये
sir.
रिकॉम्बिनेशन एनालिसिस करना है, तो हमें यहाँ पे गैमीट्स कौन से लिखेंगे पेरेंटल का एनालिसिस करने के लिए? पेरेंटल
पेरेंटल में सर ए और कैपिटल बी,
क्या?
कैपिटल ए स्मॉल बी
हम्म,
ये पेरेंटल में होंगे
गुड.
और नॉन पेरेंटल में कैपिटल ए एंड कैपिटल बी,
वेरी गुड,
स्मॉल ए एंड स्मॉल बी.
वेरी गुड. ठीक है, so you did this experiment and then you find that you know A and B are basically the recombination percentage, non-parental over total into 100, you find it's 25%. So it means the physical distance between A and B is 25 centimorgans. Why? It means out of 100, so 100 cells in those flies, in this one, in F1, 100 cells, they undergo meiosis, okay? And out of 100, in 17 meiosis, ये एक event है, ये दूसरा, ये तीसरा, gametes produce हो रहे हैं. Out of this, only 25 meiotic events or recombination events took place, where non-parental or recombination took place. Okay, recombination is always reciprocal.
Yes.
So Najma, I can see now. Najma is your TA for this course. Najma will finalize day and time with you for tutorial. Okay. And I would encourage you all to go there because this is a fast-paced course. This will go very fast. Normally what I cover in 3-4 lectures at undergrad level, because this is a master's level course, this is just, I'm just refreshing your basics so that as we move on, you know what to, I did not talk about alleles today. You should know what alleles are, okay? You should know what heterozygous is, what homozygous is, all these things. Okay, so next time when we meet, we'll solve some recombination problems so that you know how to analyze or how to draw a genetic map. Okay, clear? Surely. If there are no more questions, should I end here then? Thank you very much. Have a nice day there.